So now we're going to look at how we regulate those enzymes. So how do we regulate enzymes? It's going to be very important because, like I said, Mother Nature does not want to waste energy. And so we want to be sure that we regulate these, that we know when they are going to be turned on and off. And we also want to know what is going to cause problem for our enzymes. And that's kind of more what this slide is about, okay? So enzymes can be influenced by many things in the cell's environment: temperature, pH, pressure, salt concentration. No, maybe not salt concentration. I'm trying to think. There's more here, but the most important are going to be temperature and pH. Okay, so this can cause what we call a denaturation. Okay, denaturing of proteins, which is going to be a distortion of the enzyme's shape. And if you've had my class, and I've probably already said it a few times this semester, structure determines function, shape determines function. If we have a protein here, and this happens to be an enzyme, well, if you look at this globular protein, looks like a glob, but it does have a specific shape to the glob. Oops, that one kind of jerked over on me. All right, and so let's say one of these shapes here forms the active site. Let's just say this is the shape of the active site over here. Okay, so if that is the shape of the active site, and I change the structure of that protein because let's say I've heated it up too much. When you heat it up, it breaks the bonds. Okay, when you break the bonds, this tertiary globular structure of this protein, the secondary structure, which is the alpha helix and the beta pleated sheets, they all break down. Okay, and so most of the time this is irreversible. Irreversible. I was thinking I was saying the wrong word. Irreversible. We denature them. They are no longer in their native state. Okay, so it causes them to become unstable, or labile would be the word. And so when we break these, they're no longer functional. I don't have this nice little shape here for my substrate to fit in, so my protein does not work anymore. Okay, so the substrate cannot bind to the active site. We also have some ways of directly controlling the action of enzymes. We have inhibition, okay? And we have two basic types. We have competitive and non-competitive. So inhibition, competitive inhibition, I should say, is where two guys compete for the same seat. So I look at this as kind of like musical chairs. So if you're playing musical chairs and there's two of you and there's only one chair, you're both going to fight for the same seat, right? This is competitive inhibition. And so this is where the competitive inhibitor is going to have a similar shape, and it's going to compete for actually binding to the actual active site, okay? And it's going to resemble the normal substrate. It's a mimic. It's going to occupy the site, so then the guy that's supposed to get there and sit down can't do it, okay? And so it blocks the reaction from occurring. Now, non-competitive inhibition, this is where enzymes, they actually have two binding sites. We have the active site, and then we have the allosteric or other site, which is a regulatory site, which is where we like to turn things on and off, basically, okay? So this regulatory site, and I kind of like that name. I think that's a better name than some textbooks. The allosteric is kind of complicated, and that's great when you're in organic chemistry or biochemistry, but regulatory site is a really good name here. But I still say other site, okay? So active site is where the action happens, and this is where we want to basically turn this sucker off and on, okay? And so molecules then can bind to this regulatory site as well and basically turn it off. And that is almost always permanent. And so this shuts down the enzyme's activity. It's actually going to cause a change in shape, because when something binds, what did I say? It moves to fit in, and it's going to cause a change in shape in the active site, and your substrate cannot bind. So you, again, cannot make your product, okay? Another way to regulate enzymes is to be able to control whether or not they are made. Enzymes are recyclable, but they don't last forever. They do have to be replaced at some point. So we can actually repress or stop synthesis of a particular enzyme. In enzyme repression, the synthesis process is stopped. in one portion of the pathway. So remember, metabolic reactions occur in many steps. It's several steps together to create something. Okay. So one of those steps is going to be halted at. All right. So when the product of a pathway of a metabolic reaction builds up to a high enough level, when you make enough product, you want to stop, right? If you own a business and you are making, let's say you're making these little plastic bottles, when you have enough to complete your orders, you might want to go a little over that, but you don't want to just keep making them, because what if your orders fall off and you've made a bunch of these and you can't sell them? That's a waste of money, right? We don't want that to happen, okay? So we don't want that to happen in nature either, because remember, nature is thrifty. It does not want to spend any more money than it has to. So it doesn't want to make something it's not going to use, all right? So when the product of our metabolic pathway builds up enough to meet demand, a high enough level, right, to replace the enzymes that catalyze the reaction is going to be stopped. Okay, so if I have enough enzymes to do what I need to do, I'm going to quit making them, in other words. Now, induction is the opposite of repression. So this is when the enzymes actually only appear when needed. With repression, we're kind of making these all the time, and then when we get enough, we stop for a while. Induction is going to be for something we don't want to make it unless it's absolutely needed. So maybe it's something we don't need all the time, just in particular situations. So this is going to be something that bacteria use. So E. coli does this. All right. So remember, this is the opposite of induction. So the enzymes are actually not going to be made until the substrates that it's going to work on are already there. Okay. So E. coli uses lactose as its main carbon source, and it's only going to produce the enzyme to break that down, which is handily named lactase. So it's not going to make lactase unless lactose is present, because it can use other sugars, okay, but it prefers lactose. But if it's not there, why would you make lactase, right? So if it does not have lactose present, okay, it is not going to make it lactase, the enzyme to break it down. This is actually called the lac operon, and it is an inducible enzyme. The lac operon is actually the area on the gene. Anyway, so this is an inducible enzyme. So it keeps a microbe from wasting energy by producing lactase if lactose is not present. Okay, pretty cool. So this is similar to feedback inhibition, which we talked about in anatomy. Feedback inhibition is where the end product basically feeds back to that initial enzyme, usually the first one in the pathway, and it feeds into that allosteric or other site and turns the pathway back off. So when you get enough product, you're going to eventually bump into your original enzyme, and that's going to shut it off. And then when that product concentration goes down, it's going to want to unbind from that enzyme and turn production back on. It's like an automatic on-off switch. That's feedback inhibition, okay? That's kind of similar to these processes of inducibility that we're talking about and repression. So here are the steps 1 through 5. The enzyme is synthesized, it's made continuously until it's inhibited. Excess product is going to react with a site on DNA that regulates the enzyme synthesis and stops enzyme production. Okay, so it's feeding back. It's feeding back to the gene instead of the enzyme. That's the main difference here. So metabolic pathways, I've already said they're multi-steps, okay? There's a series of reactions. Each step has its own enzyme that catalyzes the reaction. In anatomy, we looked at anatomy 1, we looked at aerobic respiration, and we went over perhaps like the 10 steps of glycolysis. Well, those 10 steps of glycolysis, there are 10 different enzymes that go along with those 10 steps. I just didn't make you learn them. I've learned them, but you didn't have to learn them, okay? And so there's a specific enzyme for every reaction, okay? So the product of one reaction is going to be an intermediate product, so it's often the reactant for the other. So like we started with glucose, and then we converted that to what? pyruvate. And then pyruvate was actually the reactant for the next reaction, which was the intermediate step, okay? And then we were converted that to acetyl-CoA, which was the product of the intermediate step, but the reactant for Krebs cycle. And it goes on and on like that, okay? So a lot of pathways actually have branches that give alternative methods for nutrient processing. Some have a more cyclic form in which the starting molecule is regenerated to initiate another turn of this cycle, but they hardly ever stand alone, okay? And they can actually merge at many different sites. So here's some examples here. Metabolic reactions occurring in that multi-step series or pathways, each step catalyzed by a different enzyme, okay? And so you can see this shown here. You've got a linear pathway here, like glycolysis was linear. We kind of went in a straight line, right? Krebs cycle is more cyclic. Divergent is where you can go two different directions. Convergent is where you're going to come into one particular direction, like making amino acids. So this brings us to endergonic versus exergonic reactions. It's kind of like endothermic and exothermic, except in the body, we're going to say endogonic and exergonic, all right? And so we can look at this diagram here and tell a lot about these reactions. You can even, if I took away the words, you can tell if these are endergonic or exergonic because you're looking at the release of energy. So we have free energy, which is the energy available to do work, okay? And then we have the progress of the reaction. So here the reactants are lower in energy than the products. And so we've had to put energy in to the reaction. Okay. And so this is the chemical definition of spontaneous here. If it is spontaneous, you don't have to put energy in. We're putting energy into this one. And so it is not spontaneous. Okay. Energy is absorbed. So ender, think endo, inside, you're putting energy in. Exer, think exit, you're releasing energy. Your reactants are higher in energy than your products, and so you are releasing energy, okay? You're releasing energy. It is now made available to do work, all right? So these are often coupled so that energy released is immediately put to work, and this does happen in cellular respiration. All right, you've probably heard of this a little bit if you've taken me for anatomy, but just in case, we're going to go over redox reactions, which is an oxidation reduction reaction. This is where electrons are transferred, and this is a big part of our cellular respiration, okay? These always occur together. So oxidation is a loss of electrons and reduction is a gain. And that's a really important thing that if you aren't already saying it in your head and you took my class, you should be saying it. What is the way we remember this? Oil rig. Oxidation is a loss of electrons and reduction is a gain of electrons. Sometimes in chemistry they'll say one is Leo, the lion says Gar. I don't really know that one, but that's so much more complicated. Why would you use it? I like oil rig, okay? So oxidation is a loss of electrons. A compound loses electrons, it's oxidized. Well, those electrons have to go somewhere, so they're going to go to another compound that is reduced, okay? And so this is really important when we're talking about energy transformations, okay? The law of conservation of energy tells us that energy is not created or destroyed, it's only transformed. It's only changing form, okay? So we have enzymes, we're taking them from one substrate atom to another. So we have enzymes that carry things needed for the transfer of electrons, okay? And these are coenzymes, and these are our NAD, N-A-D, if you remember from cellular respiration, nicotinamide adenine dinucleotide, okay? And then we have FAD, which is flavin adenine dinucleotide. And you can do innate NAD FAD, that is just fine, okay? And so these are our little electron shuttles, is what I've called them for you, okay? So they're little electron shuttles that are alternately loaded and unloaded. They carry electrons, okay? They accept electrons and release electrons and hydrogen when we're talking about the body. And so they facilitate those redox energy transfers. In a catabolic pathway, electrons are removed and carried through a series of redox reactions until the final electron acceptor at the end of the pathway is reached. And hopefully you know or remember what that final electron acceptor is in aerobic respiration. For a eukaryotic organism generally is going to be oxygen. Now in some bacterias or in anaerobic metabolism, it's going to be another compound. Energy in the present in the electron acceptor is captured basically to phosphorylate. Big word, simple meaning. Basically, that means add a phosphate on. Okay, add a phosphate on, take a phosphate on. That occurs over and over and over again in the body. So you're going to add a phosphate group to ADP or another compound to store energy. Okay, so ADP, you're going to add a phosphate from adenosine diphosphate, and you're going to make it adenosine triphosphate, and that's going to be what we call a high energy bond. That last phosphate added on there. Okay. The cell doesn't handle electrons as separate entities, all right, but rather it carries with them as an electron and a hydrogen when we're talking about in the body, okay? So it consists of a single electron and a single proton, and they kind of travel together. And if you'll notice, there's a definition down here, dehydrogenation. And again, break these words down. What does D mean? It means to undo. Hydrogen is a process, so you're just removing a hydrogen. Okay, if you're removing a hydrogen, then you're carrying electrons with it. All right. All right, so ATP, this is our metabolic money. This is our energy currency, right? It's going to be made of a nitrogen containing or nitrogenous base, which in this case is adenine. Okay, and then you're going to have a ribose sugar here, 5-carbon sugar, and 3 phosphate groups. And remember, this is going to be a high energy bond here. Okay, so the removal of that, we would say terminal, it just means the last one. Phosphate is going to release energy. ATP is that main energy currency of our cell. If it's used up in a chemical reaction, it's got to be replaced. So this makes using and making ATP a never-ending cycle. And when I say making, we're basically talking about putting the phosphate on, taking it back off, okay? We're just transferring. We're not making anything brand new here. All right, and so you can see using ATP, we start with glucose. We've got ATP, we're phosphorylating it, we're removing a phosphate group, and we're releasing energy, and ADP is formed, okay? And so we're doing that to do glucose 6-phosphate. So we removed that phosphate group, released energy, and added it on to glucose, and actually to the 6th carbon. That's all that means, all right? And so the input of energy is needed to replenish your ATP, okay? Energy released during the hydrolysis of ATP. This is going to drive biosynthesis. This is driving your reactions where you're making something. It provides that phosphate to a substrate before linking it to another molecule, okay? It's also used to prepare molecules for catabolic reactions, like when a 6-carbon sugar is phosphorylated in glycolysis, okay? And so you're making it, you're doing the opposite, okay? If ATP is used to remove, it's going to release energy and needs to be recreated. This, however, requires an input of energy from somewhere, okay? In heterotrophs, catabolic pathways provide energy that is needed to generate that high energy phosphate. All right, so practice question here. A reaction that requires energy as it goes forward. Is it extraconic, endergonic, oxidative, or reductive? Pause. B, it requires energy. Ender means endo, going inside, so it is endergonic.